level of RUS. But when they are exposed to the hydrogen peroxide oxidizing agent, fluorescence goes up, and this fluorescence further reduces when they are exposed to the P. So P faded cells have lesser fluorescence in presence of oxidizing agent, further clearly indicate that it has got a very high anti-oxidizing ability. Similarly, we did it with the human skin microbiome. So like the same way, we could also identify. And some of the experiments which we did it at the IIR with the Professor Tiwari, and he helped us, and we got the paper also in that from that. And it's in good impact factor journals. And we noticed that, see, you can see that these are all the locomotory factors. When you have cells in a normal conditions, this is controlled without any antioxidant, without oxidant or oxidizing agent. We have and when the drosophilas are with the P, fitted with the P, you find that they have a better locomotory activity. And similarly, when they are exposed to the something paraquat, you can see that with the presence of P. And at the same time, we could also measure the what is known as, these are called superoxide dismutase and the catalase activity. So these activities will go down in presence of PE. That's what we notice. These are all the things which were. So it indicates that, and very interesting experiment which we did. There what we did, we use a C. elegans, and that C. elegans mutant is such that where we introduce what is known as A beta, that is the beta secretase gene of human, is placed in the muscle cells of C. elegans. Are you getting? The gene meant for the beta secretase, we call it A beta 142, something like that. That gene is placed in E. coli in the muscle cells. Since that gene is placed in the muscle cells, and it is placed in such a way that the promoter is a temperature sensitive promoter, heat shock promoter. So normally we allow the C. elegans to grow at 20 degrees centigrade. If I increase the temperature from 20 to 25 degrees centigrade or 22 degrees centigrade, what will happen that the promoter will become active, with the results the gene which is placed in the muscle cell will become active, and as soon as that is produced, you will find that it causes paralysis in the C. elegans. But then it will not, it will take some longer time, and that's what it noticed. So you have a control, this is in presence of P. You have a control, this is in presence of P. So by simply shifting the temperature, you are making the gene to be active, to express. And that's how we did it. And that resulted into the several, basically what I want to say that P is responsible to expand the lifespan and health span of C. elegans. It improves the tolerance against the thermo and oxidative stress. It is independent from the IS signaling, that is the insulin growth factor, and it reduces the intracellular level of that. These are all the things which are happening in this. Like that, we got, and at the same time, we are also using these. I will not discuss much on this. We are also using these for the microsporin like amino acids preparation, and they are used for variety of purposes. Again, basically our purpose is that to use this small molecule as a UV protectant. And these days we get the various lotions which are called SP25, SP50, 100, something like that. These are all the UV protectants. So similarly, we can get the molecules from the cyanobacteria, and this molecule can be used for such type of the protection. These are all, this I will not, this is what actually happens when you are exposed to the sunlight, and especially when they are exposed to the ultraviolet radiations. There are so many imbalances, and these imbalances which are happening in the cell, but then they have a various defense mechanism. If these defense mechanisms are failed, then obviously it can lead into the various complications. So that's what I was, so we try to go for that, and then these are all water soluble absorption having, like these are all some compounds which are produced, and these are all the structures which are available. Then how happened, and that's how. And a very simple explanation what you have when they are exposed to the heat, UV radiations, how it happens in the abatement factor. And these are some of the experiments where we have proved that it can give the protections to this microsporin. And one of the simple case, the morning he was talking about the other lab, and we also have prepared certain such thing like you have a, when they are exposed to the sunlight, when you use the olive oil, you will find the skin like that, commercial skin and our microsporin whatever. We have a better result in comparison to this. This is what happened. And these are all the things what we have. And this is what I am speaking, but basically I don't do any work. My job is basically to get the grant from the various funding agency, make the things available to the student. It is the student who works, and I take the credit by presenting you. So basically, Mr. Once again, thank you all. It would be my pleasure. If you have any queries, any questions, I'll try to. Yeah, please.
Thank you, sir, so much. That was very, very informative. I have so many questions, but I'll keep it.
Yeah, please. Anyway, I am there at Charu set, so
anytime. One question I wanted to find out. You spoke about the half-life of all of the various, like the C. elegans and the nematodes and all of those things, and how it has increased with the incorporation of the pigment. But my question is, what was the half-life? Were you able to figure out what was the half-life of the pigment itself, if it is unprotected against the sun, different temperatures, and all of those things?
Yeah, you're right. Very good questions. Yes. Did you understand what is that pigment which we are using it also has its own half-life. So as long as it is protected from the sun, as long as it is protected from the light, it has fairly long half-life, at least several days. But whenever you perform the experiment, these pigments are given for a smaller period of time. And secondly, they are given in a specific confined condition. That means you are putting into the beauty incubator, which is maintained at temperature 20, and it is in the dark condition. So the pigment will not get damaged during that process. So it will remain. And then these days we also have a pigment which have been not only isolated from the normal cyanobacteria, but we also got one of them is called oscillatoria, which has been obtained from the hot spring. And that oscillatoria also is responsible to produce, and we notice that that pigment is stable even at 50 degrees centigrade. So we believe that even if the temperature of the earth goes up, still we have enough time. Maybe by that time we may have some other culture also ready, but up to 50 degrees centigrade, that pigment still remains stable for several days. Absolutely, provided it is protected from the light. Yes, sir.
Sir, it was a nice presentation.
Thank you, sir.
And always, as always, you inspire us. So, sir, I have a question. Means,
yes, sir.
You've shown your results, and there are increase of oxidative stress, and by feeding the C. elegans and you have shown that it increases the antioxidant property. So, because a recent theory said that there is a problem with the mitochondria when there is aging. So, is there any study that was conducted to see the mitochondrial health after the feeding the antioxidant?
We have not done anything, but there are 2-3 things which are planned. But unfortunately, I am now at such a stage where getting the good fund from the different funding agency and keeping the students booked with that, I find it myself little difficult. Now I am approaching near 70, so my wife is also getting. I am not able, but I have 2 plans to do. One is, of course, what he rightly pointed about the mitochondria, because that is one part which is highly essential. It's the powerhouse of the energy. So if that is protected, perhaps things can be taken care. Second is that we have been seeing, because these are comparatively large protein. If you see the whole complex of any protein, the size is somewhere around 200 plus kilodalton, and such a protein will always have a barrier problem. They will not pass through the membrane. And not only that, and here in all our experiments, what we did, we gave this protein through the digestive system. And whenever you give any protein through the digestive system, obviously there is a tendency to undergo into the degradation. So it will always have something. So what we have noticed that there are one or two isolated papers, but not very clear. It has been noticed that it is not the whole protein which is responsible for that. But there are certain fragments which are mainly hydrophobic in nature and charged molecular amino acid. Those molecules are responsible for antioxidant. But then what is that fragment, we still don't know. So we have a plan where we'll do the random fragmentation of the protein, and each of the fragment, but then it is a long-term experiments. It's not going to be, because it's a lot of error and trials that have to be done, and that finds to be there. Maybe with the AI's knowledge can help us to design the experiment properly, but still we have difficulty. That is one. But secondly, what he said that, but that's again long-term plan where we can work with the mitochondria. But so far we have not done anything. This is all rough information what I can say.
Thank you, sir.
Sir,
Yes, sir.
you are using phycobilisomes.
Yes, sir. Phycobilisome is a giant molecule, sir.
And phycobilisome itself is a photodynamic.
Yes, you are right, sir. Perfectly.
So if it gets exposed to light, it will undergo state transitions. And it may produce rocks.
Wow, oxygen. Yes, sir. Yes, you are right. Perfectly.
So how it is pertinent if we are using or trying to commercialize it?
Yeah. Now, at present, our purpose is to commercialize as a nutraceutical rather than anything else. We are not at present trying to do unless and until we get a specific molecules, specific process, and then perhaps as an it can yeah, like for instance Alzheimer, it has a good activity, but they need to maintain.
No, it can be done for treating pimples.
Yeah, could be, yes, could be a possibility, sir. But then there are, for example, we say that though doctors say that whenever there is a ventilations, the oxygen is given. But there are theories also say that too much of oxygen is also dangerous. So one has to have a balance between the two. How do the oxygen, whether it's really essential or it's not, to what extent. It is like that here also, because you can see the one controversial statement in my talk also. I use the word called anti-cancer. When you say that anti-cancer, you require a oxidant to be generated so that the cancerous cell can be destroyed. Absolutely. But here I am using the antioxidant to destroy. So, but then pathways are different. They are not the same pathway where it works. These are different ways. But then it requires a lot of other components.
Thank you. Sir.
You mentioned that phycoerythrin works as an anti-aging agent, right? And sir, the aging is also related to telomere shortening.
Yes, it is very much.
Somehow affecting.
No, we have not checked. That becomes altogether different work. No,
Yeah.
we have not tested. We can't say. No. Telomeres, because you see that in case of telomere shortening, the time period which requires to shorten the it's a damn it is no, absolutely.
Okay, thank you.
I sincerely thank Professor Madambar for sharing his profound expertise, and for such skillful session, I request our POVC and Director Professor P.C. Vyas sir to present a memento to Professor Madambar as a token of respect and gratitude.
Thank you, sir. I came down to your office, but just now before coming here, we thought we'll meet you. Nice. Thank you, sir.
It was indeed a spectacular session.
Thank you.
Please give a big round of applause.
Thank you very much.
Thank you, sir. Moving to our fourth invited talk, we have Dr. Shailendra Gaur, Director Process Science Group Cipla Private Limited from Bangalore, with us for an expert talk. So before we move to our expert session, I request Professor Anand Tiwari to greet Shailendra sir with a bouquet. तालियां बजती रहनी चाहिए. Thank you, sir. Dr. Shailendra Gaur, a passionate researcher with an aim to serve science and make a positive impact in patients' life. He earned his doctorate in biotechnology from Department of Biotechnology and Microbiology, Gujarat University. Biotech enthusiast with over 2 decades of biotech industrial experience, Dr. Gaur has started his career with Torrent Research Center and worked with organizations like Reliance Life Sciences, Nicholas Piramal, Intas Pharmaceutical, Viatris Global Biologics, working in the field of biosimilar development, experienced in early stage product development like cell line development, upstream process development and technology transfer. Also worked on more than 20 biosimilar molecules. Multiples of them are registered product in global market. Put your hands together to invite a very charismatic and zestful personality, Dr. Shailendra Gaur, for his talk. Over to you, sir.
Thank you so much for the wonderful introduction, and thanks organizers for this wonderful opportunity. But I think my talk is slotted at a very wrong place, because coming to dais after someone like Dr. Datta, who has so much, who has served the science for so many years and has so many accolades in his name, it will definitely make you feel nervous, right? And I am nervous at this particular point of time. Thank you so much, Dr. Datta, for your wonderful talk. And I hope all your work translates into a wonderful product and we all have a long and healthier life to come together. Thank you so much. So as everyone is aware that I represent industry, so I'll not be focusing a lot about AMR in my talk today, but I'll be focusing more on what industry can bring to convey this, right? So we'll have a little bit of introduction about AMR and what monoclonal antibodies are, how industry can help this, and then we'll discuss about some of the areas of concern. We'll look at some of the facts and figures of AMR and how industry can help, and then I will quickly summarize my talk. So I have some time, right? Okay. Fine. So we know in the past we have developed a lot of antimicrobials like antibiotics, antifungals, antivirals, which can be used to fight against antimicrobials, but in certain conditions these microbes develop the resistance against these drugs, and then this AMR or antimicrobial resistance comes, right? And this drug resistance make all these antimicrobial medicines ineffective, and that leads to further infections and further deterioration in the quality of life of the patient, and it also has a huge financial burden, which we will be looking at in the coming slides. So let's quickly go through some of the major causes. One for sure is misuse and overuse of antibiotics. The conditions are like when antibiotics are used inappropriately, or they are prescribed wrongly against an infection. You must be aware that in Western countries it is impossible to get an antibiotic over the counter, but in India that's not the case, right? थोड़ा गला खराब है, एजिथ्रोमाइसिन ले लो, वो काम नहीं कर रही, एमोक्सिसिलिन ले लो, and everything over the counter, which is actually the main reason of doing this. Other reason, as Dr. Datta has rightly mentioned, these microbes have slowly genetic changes, phenotypical changes, through which they develop this, because they are also a living organism. You provide them an antibiotic or antimicrobial, they also want to survive. So they will find their own ways by doing certain genetic changes or developing some phenotypic characteristic by which they can show the resistance to these drugs and can survive, right? So that is another reason. One reason that was a surprise to me when I was also preparing this is lack of new microbial programs. And if we believe WHO, and we have to believe WHO, when we believe that they clearly say that from 2017 to 2021, there is a decrease in the new antibiotic development programs in clinical trials. That's a scary news. And the last class of antibiotic that we have developed was late back in 1987. So since then we have this huge discovery void. We haven't introduced any new class of antibiotic or any antimicrobial after 1890 or 1987. So this is a huge void, and there is a dire consequences for this, which we can talk later, but we need to work around this, right? So what are the consequences? Once we are talking about what is AMR, but what are the consequences? One for sure is the public health. Because of this, you have increased suffering and increased death in the patient. They have prolonged hospitalization, multiple treatments to treat these diseases, because of which they have huge side effects. Then at times it becomes extremely challenging because of these antimicrobial resistance organism, that the treatment becomes almost impossible, and there is no way but to see the patient go, right? Apart from the public health, it has huge economical burden, like more hospitalization, more drugs. There would be huge cost on the patient, and to develop such programs against antimicrobial microbe, you need to have a huge plan, huge investment. So that's a financial burden on government also. And a threat is that it has been discussed, it has been noticed that by 2050 there would be almost 1.1 to 3.8% of GDP loss because of this, and the amount, if you see, it goes in multi-trillions of US dollars. So which is huge for economic burden. In one place we are seeing of developing nations, we are seeing on developing the world, and if we have certain conditions like this where our GDP or revenues are going down the drain, it is very stressful situation. And these antimicrobial things does not just affect the human, but they also have huge impact on crops as well as livestock, right? Because with this antimicrobial condition, your crops or your final food will become contaminated, which will give you infection. The livestock, the animals, they can also get infected with these, and they cannot be then used for the purpose that they have been used for. So there is a huge issue. Let's check some of the facts of the health card. It is said that close to 5 million people die every year because of AMR or AMR associated diseases, out of which 1.27 is directly through AMR. Then the sad part is, out of every 5 patients dying of, 1 out of every 5 patients dying of AMR is a child who is less than 5 years of age. So if we are not able to cure this, just look at our future. And without preventive measures, it is said that by 2050, this could be the first reason of death in the globe, right? At present, this is stated at the 3rd cause after your heart diseases and stroke. Then comes the respiratory diseases. And if this went uncontrolled, then this can take the first place, and which is really sad part. Look at some of the data from domestic circuit as well. In India also, this is the 3rd largest cause of death, and in 2019 close to 3 lakh deaths were attributed to AMR, and close to 10 lakh deaths associated with AMR, like AMR and secondary infections of that. So that's a scary figure, right? And these are the 5 pathogens that are the main culprit in Indian scenario. Going further, as we discussed, that we may have a loss of 1 to 3.5% of our GDP. Now this is an interesting figure wherein the author has compared the 2009 financial crisis to what it could be because of AMR. So this is normalized against the condition of before the financial crisis. So here you can clearly see that the loss that was taken was close to 1.7 to 4% of loss in GDP, and this is the overall world's average. Now you can see these colors. The black one is coming from low income group, and the orange one is coming from the higher or middle income group. Now during this financial crisis, we could see the low income countries were affected minimally, right? But that is not the case in AMRs. If we have a AMR which is uncontrolled, the least income group countries will suffer the most, because they don't have infrastructure, they don't have medicines, they don't have proper health care, and this will lead to further increasing the gap, the economic inequality between these countries. So this is fearful. Now, as we suggested, because of this loss, we can lose almost up to 100 trillion USD every year if this goes uncontrolled. What are the current approaches that different government are taking to combat this? One definitely is the antimicrobial stewardship programs, which is nothing but you have coordinated programs to promote appropriate medications. They train physicians, they train patients what prescription to be done, how, and to patient that how this prescription have to be taken. You have to take it only from a licensed practitioner and all. Then you have surveillance and monitoring. What you are doing in this is whenever there is an incident of antimicrobial infection, they are surveilling it, monitoring it, and tracking the infection, so tracking their spread so that they can combat it. And then another thing is they are giving incentives for developing antibiotics, newer antibiotics, which can go in the terms of say some market reward entry, some relaxed regulatory environment, so that multiple companies can put their money in this. But is this enough? And if this is not enough, what else we need to do? Which area we need to look into? According to me, these programs are not sufficient to take care or combat AMR completely, right? We need to look beyond. And when we say we need to look beyond, what are the areas that we can work around? Normally, whenever we have an infection or any antigen that is entering our body, our defense mechanism or our immune system has its own way of treating that or its own way of removing that pathogen, right? Which is through antibody development, right? So whenever a pathogen or antigen enters your body, your immune system gets triggered, it produces antibody which is specific to that particular antigen, it binds to it and through different mechanisms it cleans it off, right? So can we go back to that? How? I'll explain you in the coming slide. So antibodies, as we just discussed, they are normal Y-shaped structure. So there are two figures, one is a little simpler one is a little complex one, the ribbon structure of the monoclonal antibody or the antibody. So antibody is nothing but a polypeptide of 4 chains, 2 identical light chains and 2 identical heavy chains. If you look at this structure, it is further divided into a constant region which is reserved or conserved against species. Say a mouse will have a fixed constant region, human will have a fixed constant region, and then you have a variable region which actually gets changed every time a new pathogen enters your body, right? So this is how this antibody is made, and in normal conditions, whenever we have infection, your immune system gets triggered out and produces these to eradicate this. So can we somehow use this technology in spite of using the serious antibiotics which have side effects and all? Yes, we can. We'll go further to discuss about what are monoclonal antibodies. So the name suggests that it is mono and clonal. Whenever we talk about clone, it has to do something coming from a single cell, right? So to define this monoclonal, it is a antibody against a specific antigen with a single epitope recognition site coming from a single cell. Again, means as we are talking about normal antibodies, it is against B cell. So it has a specific epitope, right? It shows specificity to a actual or single epitope and it clears that. So that is monoclonal antibody. But in general, in normal situation, in vivo conditions, you generally generate polyclonal antibody, because when a large size antigen enters your body, you suddenly trigger an immune system, and there are different B cells which recognize different epitopes on that antigen and produce antibodies. So in normal conditions, in vivo conditions, you generally come up with polyclonal antibodies, but in industrial setup, we can generate monoclonal antibodies which are specific to a antigenic epitope. Why do we need this? Because we don't want crosstalk or off-target effects, and that's why monoclonal antibodies are extremely important. The first monoclonal antibody was developed close to more than 40 years back, which was OKT3 or anti-CD3 antibody, which was used in graft rejection during kidney transplant and all, right? So this was made using a technology called hybridoma. And since then, maybe this number is a little conservative, maybe more than 90 monoclonal antibodies since then have been approved by regulated market like US FDA, EMA and all. So if we talk the same thing when we talk about monoclonal and polyclonal, through this picture it would be a little more clear. So this is the antigen that entered your body. Now all these B cells will start identifying different epitopes on this, and these cells will start generating antibodies. Now when you take all these cells in one go, produce antibodies, then it would be called as polyclonal, because this will have specificity to different epitopes. Now when you isolate, say, a single cell from this pool of B cells, grow it, isolate the antibody which has only single specificity to a specific epitope, then this is called a monoclonal antibody. So how is this developed? Or before this we'll just discuss about the advantages. Most of them we have already discussed. So one is definitely the high specificity. It will be extremely specific. The monoclonal antibodies does not bind to any off-target effect. Right? So using this high specificity, we can develop these antibodies against a specific epitope on any of the pathogen and can use this. There is extremely low risk of resistance development, because as I said, this is again part of your normal immune system strategy, right? So you're developing the resistance against this would be a little hard for the microbes. Then one potential area where MAPS can be used is the combination therapy. Now we know normal antibiotics may not be that effective, but if we combine them with certain monoclonal antibodies, the treatment would be even more effective. So this is one area where a lot of research is going on, and there are some significant development in this area. Another thing is looking at the numbers of cases that we are seeing now. We need a technology that is scalable. We cannot always keep producing that on a small scale. And when we talk about monoclonal antibodies, the recombinant DNA technology that has taken the advancement, now we can grow this at as high scales as 50,000 liters or something. So from a single batch we can produce enough material which is sufficient for sufficing a lot of requirement. So scalability is again a very positive impact of monoclonal antibody. So how do you make an antibody monoclonal? So as we said, the first and the oldest technology to produce monoclonal antibody is through hybridoma. What do we do in hybridoma? So hybridoma technology was developed in 1975 by these three handsome scientists who has got Nobel Prize for this work. And what they did was this is nothing but a fusion technology of two different cell types, trying to utilize the characteristics of both these cell lines to get a cell, a fused cell, which can produce antibody for a longer period of time. Right? And this is the pictorial representation. What you do in hybridoma is you induce or challenge a mouse with the antigen, required antigen for which you need antibodies. Now it will start secreting antibodies, and you isolate their spleen cells. Spleen cell is where you get your activated B lymphocytes, which is responsible for producing antibody. Now the different color shows the different specificity of the antigen. So each cell may have a different specificity for that particular antigen. So when you combine them with the myeloma cell, you will have a hybridoma which can now produce antibodies, and then you isolate them individually, pick up a single cell, grow them under selection pressure of hat media. I'll explain you what this hat media is, and then you, whatever the population you get, that actually is producing a monoclonal antibody. Now how does this work? We know spleen cells can produce antibody, as we have just discussed, but as they are primary cells, they cannot be grown for a longer time. They'll die off relatively soon. On the other hand, myeloma cells, which are cancerous cells, can grow indefinitely. You can grow them for, they are immortals, and you can grow them for a longer time. So can we do something to utilize both these characteristics from these two different cells? Yes, we can. What you do is you fuse them using PEG or polyethylene glycol, and then grow them in a media called het media, which contains hypoxanthine, aminopterin and thymidine. Now what this does is, for nucleotide synthesis, every cell requires nucleotide synthesis for DNA replication and all. So there are two different pathways, de novo and salvage pathway. So aminopterin block those enzymes which are involved in de novo pathway. So if a cell is put under het, they have to go through salvage pathway. Now these myeloma cells do not have enzymes required for nucleotide synthesis using salvage pathway. But your spleen cells can express those enzymes. So what this technology is doing, it is pushing your cells to use salvage pathway so that spleen cells can be used, and from myeloma cells it is taking the immortality, right? So now they are fusing them, and only those cells, now it is a fusion reaction, uncontrolled reaction. You may have fusion cell between spleen and spleen, you have fusion between spleen and myeloma, you have fusion between myeloma and myeloma. But due to this particular selection, only those cells who are rightly fused with spleen and myeloma, only those cells will survive, because it is taking some characteristic from both the cells. And then these cells, whatever has been recovered from this, you isolate them in single cell, because you need a monoclonal antibody, a single specific antibody, and then survive this. So this is the technology that was developed long back, but then since then in last 40 years there is a huge advancement. But if this technology was already there, what was the need to go beyond this? And the reason is immunogenicity. Now these antibodies, as you have discussed, are taking spleen cells from murine origin, they are taking myeloma cells from murine origin. So whatever antibody is made, that would be completely murine, right? And you must have heard about HAMA response, human anti-murine antibody response, right? What it does is, because every enzyme, as we also discussed during Dr. Datta's presentation, that every species have their own trademarks or some signatures. So in this also we can clearly say that in some post-translation modification, certain enzymes or certain modifications are species specific. So in this there are certain modifications which are not found in human but are present in murine. So when you put this antibody in you for human use during diagnosis or during the treatment, your immune system again will consider them as a foreign object, right? While it is doing its work of clearing of your antigen, but as your system is considering them as an antigen, what will it try to do? It will try to clear this off soon. So you may have a response for a lesser period of time, for which you need to increase your doses, which we don't want, right? So in the last 40 years or so, there has been tremendous advancement in this. So earlier it was through hybridoma, so 100% murine, so had highest immunogenicity. Then a second class came which was chimeric, wherein the researchers have just replaced the constant portion from human and variable portion from murine. So it was close to 65% human. It has little less immunogenicity than complete murine, and so on. Then they went to humanized, wherein they just kept the CDR portions of murine and rest everything, 90% of the antibody was human. And then finally the latest version is fully human antibodies, wherein they have completely replaced those genes in either transgenic mice or something, and have modified using genetic engineering tools, so that even if there is a transgenic mice or cell lines isolated from them, they can produce a complete human sequence. So this is the advancement in these monoclonal antibodies, and whether it is a chimeric or humanized or fully human, you can identify them through this suffix. Like rituximab is a chimeric antibody which is used against certain cancers, then trastuzumab, zumab, which is used for breast cancer and all, then adalimumab or umab. So these are suffix which you can use to identify whether this antibody is chimeric, humanized or fully human. Now another advancement, so all this thing is fine, but how to produce these antibodies in vitro? And the answer for this is recombinant DNA technology. In recombinant DNA technology, what are you doing? For all these specific antibodies, you need to first identify the sequence, as Dr. Datta has mentioned that for those for those proteins that they have isolated, they went back to see what was the DNA sequence, then they codon optimized that for a better expression and all. So that was reverse engineering of this. The actual process or the normal process for this, you have a DNA. You put this into a cell using a plasmid, and now you ask your cell to produce that cell. Now there are many steps involved here, identifying the sequence, codon optimization, and a lot many things to get a proper expressions. Once this cell line is done, then you put up in production so that you can deliver a lot of antibody in a quicker time, which is in a completely controlled way. And apart from your gene of interest or your protein of interest, it also produces many proteins. So CHO cells typically produce more than 100 different proteins, right? So you need to purify your specific protein using different chromatographic applications. There are different unit operations, and once that is done, you formulate into a proper drug. In formulation, what is the main? There was one question that what is the stability of the product. So the formulation is generally done to increase the stability of the product, right? Protein is there, but if these are not the conditions, or these are not the drugs which are made today and used tomorrow, right? You need to go through a complete cycle, so you need to show stability of these products. Say if you are doing a clinical trial which goes for around 1 to 2 years, you need to show that your product is stable up to that time, right? And beyond that. So formulation is generally done taking care of stability part of your final drug. So it has to start somewhere, right? Recombinant DNA. The first step is the cell line development. I always try to keep this because this area is very close to my heart. Cell line development is where your actual recombinant DNA technology or the monoclonal antibody development starts. So what are the crucial aspects of an ideal cell line? It should be clonal because you need a specificity and it is a regulatory mandate. It has to be stable, and this stability is not for the final product but your cell line, because when you clone this or when you make your first cell line, you do it in laboratory makeup, right? But that is not the place where you will produce your final drugs. You will produce your final drugs at a much larger stage, wherein you need to grow them to certain passages to reach to that scale. Then there would be production run. So your clone should be stable to get through that scale up. It has to be highly producing because every company wants to produce a commercially viable product. So it has to be commercially viable and desired quality. This is one of the most important aspect. If you are producing a monoclonal antibody, if it does not have a right kind of post-transcript modification or refolding or so, it may not have its proper bioactivity. So quality is one thing that is most important, and it should be scalable. As we said that the scales that we use to produce commercial product is not 1 or 5 liters, it goes into thousands of liters. So it has to be developed in a way that the process is scalable to higher levels also. And this is just overview of again cell line development. How does it work? It starts with sequence identification, codon optimization, and vector construction. Once that is done, you introduce that into a CHO cell, screen them at different stages to make sure that that is a clonal population. You screen them for quality and titer. Once you are sure that you have found a proper clone which is producing your desired product with a desired quality, you bank them, because this banking is extremely important because this will remain till the product life cycle of your drug. Then once the cell banks are done, then you start your process development and take it to higher scales. Now all these antibodies, whichever has been approved and developed, are more than 95% of these drugs were developed in a cell line called CHO cell line or Chinese hamster ovary. There is a very interesting story how this CHO comes into picture. It is being used throughout the world for all genetic application, for industrial purpose and all. But the story was very interesting. So this started during 1948 Civil War of China. As the name suggests, Chinese hamster ovary. So these cells or these animals, hamsters, were only found in China. So there was a group who was led by Dr. S.C. Yu in Nanking. Right now it is called as Nanjing. So they had this laboratory where they were breeding these animals and all. There was another American scientist, Robert Watson, a physician who was that time working on malaria in China. He requested few animals, and this was, as I was telling, this was at the time of Chinese Civil War, where communists were going to come into power, and they were sure that if that happens, it would be impossible for them to take out these hamsters out of China. So against the suggestions of American embassy and his friends, he dared on a night of December 10th during that civil war to draw a vehicle in incessant rains from Nanjing to Wuxi and then to Shanghai to put them into the last flight which was going to USA. And ultimately both these, Dr. SCU and Dr. Watson, were charged with war crime because of this activity, but they have actually served the science by doing that. So what all work we all are doing, we are actually doing on smuggled cell, for your information. So once that is done, so this is one of the I would say a script of his own diary, wherein he says this is for 11th December when he drove down to Shanghai, and he was just checking these hamsters, and he's writing couple of things I would read from this. So the hamster made the trip just fine and weakened with the chattering. They woke him up, and he's saying that juggling by the way that they ate black beans and millets and chattered all night, they are looking fine. He termed them as they are cunning little animals, right? And that's how he brought them. But story doesn't end here. So he brought it to USA, and they tried to make them survive in American condition for a long period of time, but they were not successful. Then there was a breeder who was known as magical breeder for animals, Mr. Victor, who breeded them and made a library out of it, and from there he started supplying. So the first cell line, if you have read about Cho cells, then Dr. T. Pluck, he got one female and one male Chinese hamster from his library, isolated ovary cells, and from there the journey of Cho cells in industrial use has started. This is the typical way how they look as adherent, but in industry generally they are made to survive in suspension culture because of scalability and all. So there are lot many stories like this, and we all are aware of photo 51 or picture 51 of DNA, right? The picture was taken by someone else. The picture was again without her information transferred to someone else who got the Nobel Prize. So there are so many murky stories in science, but everything ultimately I feel have served the science to a certain extent. So we need to forward those black paths. Fine. So what are the how these industrial solution can help in AMR? So as we talked about these monoclonal antibodies, definitely they have high specificity and can target the microorganisms directly. That is one application which we can use as. Other and more important or more high used application of this is combination therapy with other antibiotics or other antimicrobials. So they have shown tremendous effective effect when they are used in a combination with the traditional drugs. And we all know that looking at the potential of this, going forward they can also be used as prophylactic. Let us have a look at some of the case studies. MRSA, your methicillin resistant Streptococcus aureus. This infection is hugely affecting public health, and more than 80,000 infections and close to 11,000 deaths are happening only in US because of this. There is huge research work going on this, and people have identified different monoclonal antibodies which can be used against MRSA in the treatment. So there are multiple antibodies which are in clinical trials and are showing very significant results. Another example for this is gram-negative bacteria. We know gram-negative bacteria are involved in numerous infections, be it bloodstream infections, UTIs, surgical infections, pneumonia and all. And according to these studies, it is having a huge financial burden because cumulatively these diseases are huge. And other aspect is these gram-negative bacteria have a typical cell wall because of which they generally generate resistance to these drugs. Again, a lot of studies, research is going on to come up with different monoclonal antibodies, different combinations of monoclonal antibodies against gram-negative bacteria. and they are also in very advanced stages of clinical trials. Now, it's not just the research going on, but these are the FDA approved monoclonal antibodies against these multi-resistance, multi-drug resistant microbes. Few of them are bacteria, few of them are for viruses, including HIV and Ebola viruses as well. So these are approved antibody which says that this is being used now as a prophylactic and has potential impact on AMRs. How can we forget this tragic pandemic that we all faced? There is a huge research that was put into place during this pandemic. There are different papers wherein people have used multi-specific, bi-specific antibodies against different targets of the virus. On the receptors, they have used cocktails of antibodies wherein they mix separate antibodies which are specific to the virus epitopes and the receptors which is used to internalize into the cell, and they got huge success. This is the list of antibodies that those have got emergency use authorization for such pandemic against COVID-19. So this was developed so fast. Obviously that time the regulatory agencies have given them that flexibility, that relaxation to develop, because there was a dire need of these drugs. So these are all monoclonal antibodies that were approved by different regulatory agencies for emergency use authorization. So which shows a potential future for these monoclonal antibodies against these microbes. And this is the chart which is showing the number of antibodies in different clinical phase. Some of them are first phase, third phase, or even fourth phase, means market authorization and post vigilance. So the future is looking extremely good. But getting that antibody developed, getting that idea developed, everything has to go through regulation, right? What are the regulatory consideration, or what are the pathways through which we need to go for approval of a certain drug? So brief outcome, I know you guys are more aware of this. So under Government of India, you have Ministry of Health and Family Welfare, Ministry of Science and Technology. Under Ministry of Health, you have SEDESCO and DCGI, who are the apex body who regulate this and give the final authorization for these drugs. And then under Ministry of Science and Technology, you have DBT, which oversees these development activities and recommend RCGM for suitable actions. Apart from them, you have different regulatory bodies which works in different capacities and help us develop this overall product. You have RCGM, we have just discussed, which monitor the safety related aspects, which develops guideline and give the final approval for market authorization. You have Genetic Engineering Appraisal Committee, which approves the activity that are ongoing in different institutes and research activities. But how does this work? So to start your product development, to start any activity, you need to have a approval from DBT through IBSC. Once they approve this, okay, this is my product, this is what I want to work on, these would be the different organism I'll be using, these are the protocols that I'll be using. After seeing all these things, they develop this. Then once you do preliminary development studies, you have some characterization studies wherein you know that, okay, my monoclonal antibody or my therapeutic protein is the protein that I wanted to produce. It is giving those characteristics, it is doing what it is supposed to do. You do that using physicochemical characterization, you do using bioassays and all before going to animal toxicity studies. So before going to animal toxicity, you should have a good package, a solid physical characterization data and good bioassay data to show your molecule is working. Once that is done, you go to RCGM to get an approval for your animal toxicity studies. Once that is done, and this study has to be done in GLP accredited labs, once that is done, you again go back to RCGM, you submit your protocols, you submit your reports for toxicity studies, they will evaluate this in combination with your physicochemical characterization data, and then they finally give permission for clinical trials. So clinical trial starts in phase 1, phase 2, phase 3 and phase 4. So for clinical trial, you need to submit the protocol and dossier, wherein you show the complete characterization, you show the complete data from your toxicity studies and give the protocol for your clinical trials. Looking at all those data, they will see and approve, DCGI will approve your clinical trials. Once that is done, then you go back to DCGI, submit your clinical trials data for an permission for market authorization. There you need complete CT data, your manufacturing licenses, they will do the inspection of your GMP facility to see if everything is quality compliance, and then they will give permission for marketing your drug. Once the drug is marketed, another important part is, because until the drug is reached to the market, it will only see certain number of patients, right? So post-market surveillance is extremely important, because that is not the number which is sufficient to come up with all the off-target effects or any other condition that patient may have. So you need larger number. So this post-marketing surveillance generally happens for 4 years, which is very tightly controlled and overlooked by regulatory agency, but this goes beyond that as well. So in this, every 6 months, a safety reporting has to be done by DCGI for earlier 2 years, and then every year. And any process change, so today you have developed a process, certainly suddenly you find that okay, a specific vendor has stopped supply of some material, you need to change that. But all those changes, or if suddenly you start seeing impurity in your product, you need to change certain raw material, or you need to change certain protocols. But you cannot do it by your own. You need to first intimate the agency that okay, this is what we have found, this is what we want to do. You need to have a proper risk assessment, that if I change this raw material, if I change this unit operation, what could be the risk involved in this process, and what are the ways I am trying to mitigate them. Basis to that, you can make these changes and again do consistency batches to show everything is fine before releasing the material for patient use. So this is the overall pathway for any regulatory approval. Case to case basis, there would be certain deviations. Like in COVID, we have seen that there were many studies that were relaxed, and there was a fast track approval. In some cases, when you develop a specific antibody or develop a specific molecule where you do not have sufficient patients also, in those cases either you get a waiver, or you can get away with a smaller size or truncated clinical trials. So all this depends totally on case to case basis. Then what are the future directions? Future looks pretty bright for this. As we have discussed multiple times, in future I think the combination therapy will work, combination therapy with traditional drugs will work wonders. And this is the best probable way of treating these infections effectively and quickly. Then we also need to put a lot of emphasis on advancement of this mAb engineering. Right now we are discussing on monoclonal antibodies, but we know in market there are multiple other areas like bispecific antibodies, then you have ADCs, right, which are conserved. We have seen during COVID, every 15-20 days or in a month we were having a new variant, because viruses are very prone to change their genetic makeup because of these treatments and all, right? So in certain cases we need to ideally find some, because it will not change its complete structure, it will not completely change its genetics. So we need to study on their conserved regions which we can target and make a effective therapy against it. So those areas are something which we really need to look into. In the end, I would like to conclude what I could learn during this presentation preparation. So, of course, AMR is a growing concern in healthcare, and addressing this is important for patient safety and related economic burdens. Effective strategies for addressing AMR include responsible antibiotic use. This is the first step. We should not think of, okay, we'll directly take, if we develop AMR, we'll take some monoclonal antibody. The first step comes here. We need to be extremely responsible as a system, not as a patient, but we need to be responsible on using these antibiotics. Again, mAbs have shown promising start, but there is a long way to go, and we need to keep working on this, engineering this, finding new targets, working on antibodies which can work on multiple targets at a time. So there is a long way to this. Again, this is fine. The most important part is researchers, institutes, universities and government, we all have to come together to work against AMR. This is one thing that during my say industrial experience of last 20 years, this I feel I may be wrong, this is my personal view, that there is a huge gap, right? Our universities, our researchers are doing some wonderful work. Since morning I had 2-3 interactions with different faculty members, and the work they are doing is tremendous. But how many of them get translated to a useful product is something we have to work around, and their industry, university and government have to come together to deal with this. That's all from my side. Thank you so much. I would be happy to take questions if there is any.
Thank you, Dr. Girard. That was very informative. It was very interesting to see how you spoke about the therapeutic possibilities of MABs against antibiotics. But my question is related to how it can be used against antimycotics, like antifungals or anything like that, because that is also an emerging AMR that we have to basically look at or think of at least.
Right. So I have majorly talked about bacterial antibodies because this is the current area where a lot of research is going on. There are a few studies or few examples which are also shown against antivirus. So the treatment may not be different for these, right? In antifungal infections also, how the disease happens, either the fungal itself is toxic or it is releasing some toxic material which is causing the disease. So the development part of that MAB has to go that way. So if it is producing a toxin, you need to develop antibodies which can bind and neutralize those toxins. So the development part is same whether it is antimicrobial, antibacterial, antifungal or antiviral. The only thing is you need to identify those targets which you want to target using MAB and then work accordingly and develop your molecule.
So do we have any more questions? Okay.
Maybe can I just...
Yeah,
it's
definitely,
not a,
sir, definitely.
Thank you. It is not the question, but maybe it's a sort of a, you can say, what I was trying to understand that you rightly pointed out that almost about 2007 onward, there has not been new antibiotic, or even that way earlier to that also. We could not get any new antibiotic. The reason of not getting new antibiotic is that perhaps the people are not trying to develop any antibiotic, or they are not going for any modification of the semi-synthetic antibiotic and other thing. The reason is the pharmaceutical industries. Why I am saying pharmaceutical industries? Because if you are a BP patient, you require to take drug for the whole life.
Yeah.
If you are a diabetic person, you require to take a drug for the whole life. The pharmaceutical companies would prefer to produce those products which is going to have a long-term market. Whereas antibiotic, you will give it for 5 days, the job is done. So there is no long-term market availability, and it is always a race between any organism, between whether you talk bacteriophages, whether you talk about bacteria human or whatever. It is always a race between them, and in the race whoever do better will survive, other will die. That's what happened.
Right,
so
sir.
one of the major factors which coming into the antibiotic or AMR is that we are unable to produce the new antibiotic. And if I am not wrong, perhaps recently there was news, Hochard has come out with some 3-4 products which are semi-synthetic. And now government has taken the initiative. For example, Zydus is now coming out with the pharmaceutical active ingredient, they call it word, and which has been going to be in Vadodara.
Right, sir.
Yeah. So I was just trying to see that perhaps that is one of the major reasons also why the antibiotic resistance is not being taken care.
Right, sir. I totally take this point. Being a commercial organization, any company would want to invest in an area where they can have return of investment fair quickly and huge number. As you rightly mentioned, when you have antibiotic infection, when you have antimicrobial infections and all, the treatment is extremely short. Industry would definitely want to do that. So that's where the role of government comes. So as we all also discussed that government have different plans where they reward industry who put their efforts in developing these antibiotics or antimicrobial drugs. So yes, I cannot deny that fact that being a commercial organization, their main objective is to earn, but somewhere they also need to keep a point that they serve the society from where they are earning. And I would, being a part of Cipla industry, I would want, means everyone is aware of this, that this is the first drug that they launched in US, which is for HIV. So that time the treatment cost for HIV was close to $93 per week from the drugs that they were buying from US market. Cipla, when they put that drug, they sold it at $1 per week, right? So I'm not trying to boast this, but definitely like this, every company should take those initiatives where they also keep certain time and certain resources for such purposes, where they just don't look at the commercial aspect but also look at how we can serve the society.
Yeah.
Do we have any more questions? No?
Thank you so much.
Thank you, sir. This was our first industrial expert session of this meet, that is 2-day meet. And Artemis express wholehearted acknowledgement to Dr. Gaur's sincere effort and full enthusiasm. For such an enthralling session, I request Dr. Satyendra Mishra to present a memento to Dr. Gaur as a token of love and gratitude. Let's put a huge round of applause. Thank you. We now have reached to our 5th invited industrial expert talk. However, this is our last plenary session for this Artemis 2024. And for this, I invite Dr. Jinal Thakkar, Product Specialist, Metso's Ozone Biomedicals Private Limited, Haryana. I request Professor Anand Tawari, sir, to greet Dr. Jinal with a floral bouquet.
Thank you.
तालियां बजती रहनी चाहिए। इसके बाद हमारा लाइव सेशन है। Thank you. So Dr. Jinal Thakkar is a techno-commercial professional working as a product specialist for a range of products at Gujarat level in MedSource Ozone Biomedicals Private Limited, India. She has completed her doctorate degree from the Department of Biochemistry, MS University, Baroda in the year 2023. She was invited by Taiwanese Ministry of Education for the research internship at Kaohsiung Medical University in the year 2020. Her research interest is focused on developing novel methods for the detection and removal of various target analytes. She has published and presented her research work in various international platforms. She has also applied for an Indian patent. She is a lifetime member at SBCI, the Society of Biological Chemists India. With this brief introduction, may I now request Dr. Thakkar for the technical talk. Over to you, ma'am.
This mic or that? Hand mic is fine for me.
Good afternoon, everyone. Yeah, this is my first talk after my PhD. Yeah, so I'm extremely excited to be a presenter or speaker, you can say, right? And as a commercial person, this is my first talk to launch my product, or say just prove my product as a proof of concept. As a part of a friction of my thesis, I'm supposed to present here, and it was totally done by my junior, MSc junior, who was doing dissertation under my guidance, and this is how it comes to a proof of concept to my thesis, and now I'm supposed to present it over here. Yeah, so the topic is molecular imprinting of Staphylococcus aureus in polyacrylamide gel slab. for detecting the presence of the bacteria as a contaminant. As sir said that S. aureus is the most prevalent contaminant and it is showing more and more prevalence as much as we are facing the resistance over the antibacterials, right? So why Staphylococcus aureus? So Staphylococcus aureus is food poisoning, skin infections, may cause bone and joint infections. Sometimes it may cause high fever, calling bacteremia, and it may sometimes cause to death as well, right? So frequency of the infection is 100 in 10,000, and numbers are increasing due to antibacterial resistance, as I say, right? So this is the gram staining of S. aureus that was performed in our lab. Staphylococcus aureus are rod-shaped, as we know, right? Gram-positive, cluster-forming coccus, non-motile, non-swarming, and facultative anaerobes. So coming to the objective, why we have focused on imprinting method rather than developing or improving the conventional methods, right? So which are the conventional methods? These are the conventional methods: culture colony method, immunological methods, nucleic acid based techniques, or physicochemical methods. So which are the limiting factors which are hindering for their sensitive detection or can say sensitive removal of the contaminants? Coming to the culture colony method, it is very time consuming, as we know, right? Continuous detection is not possible. It may require 2 to 3 days for the incubation, and the method is not portable at all, right? After that, when we come to ELISA or IMS method, it is again time consuming, it is lengthy, interference due to labels is the possibility over there, it is not reusable, it is not portable, online detection or say continuous detection is not possible over here. Now coming to the PCR based techniques, so again the limiting factors are they are time consuming, right? The technique is expensive, it requires pure sample, it is not portable at all. Now coming to the fourth technique, where it is physicochemical method, say spectroscopy or chromatography or any such techniques. Here interference due to other species may occur. One more is time consuming. Third is it may be expensive, or it may require technical experts, and it is not portable at all, as we know. So what can be the possible solution for the same? So this is very basic and I would say cost effective method that is developed in our lab, and any science graduate can understand having that logical science mind. So what is the possible solution? Here we can say development of cheap and rapid diagnostic biosensor. What is biosensor? Biosensor is an analytical device which can sense the presence and concentration of biological analytes, right? So which are the things that can be overcome, which were the limiting factors in traditional methods. So here we can say that biosensors are very rapid, they are cost effective, they are portable, and continuous detection or online detection is very possible. Right. Now coming to the basic structure of a biosensor. So which are the components of a biosensors? So here we can say that a recognizable element is there, which will bind with a sample analyte, and it will produce a signal which will be sensed by transducer, and it will be then forwarded to the amplifier, and it will finally give the signals, and you can have the detection of particular analyte through the system. Which can be the analytes? Analytes can be antibodies, antigens, proteins, bacteria, DNA, RNA, heavy metals, anything can be there, right? And which can be the recognizing element that we are supposed to fabricate on a particular system? That may be antigens, antibody, enzymes, proteins, nucleic acid, cells, or sometimes imprinted synthetic materials like polymers, right? Which are the transducers? These are the means via we can see the changes in the signals. So this can be electrochemical, optical, mass change, calorimetric, and then we can see the output as a change of the signal. Why we are focused so much on developing cost-effective biosensors? Due to its commercialization. Here we can see that global biosensor market is supposed to reach by $32505 0.14 million by 2025. So it's a huge market potential over there. And which are the key players globally? Right. So here we can say Siemens, Abbott, Life Sciences, Johnson & Johnson's, Nova, Medtronics. These all are the key players which are prominent in biosensor industry. And here the India stands, which we can say that 30% of the global market is occupied by the Indian country. Right. So, which are the factors affecting biosensor performance? If we want to develop a cost-effective method, or if we want to improve a method, so which are the factors which we are supposed to focus upon? Yeah. So here we can say what kind of modifications can be adapted to develop more efficient biosensors. So answer is playing with these factors that can lead to more efficient biosensors. So which are these factors? These factors are improvement in biological receptor. So it may affect sensitivity, selectivity, or it may improve the both, right? Immobilization method, when we are supposed to immobilize particular receptor, right? So that can be entrapment method, adsorption, covalent attachment, microencapsulation, and cross-linking. Now coming to the transducer, if we change the transducer, it may also affect the sensitivity. Suppose say if we are using electrochemical and then we are supposed to switch to optical, then there will be a chance that we may lose the sensitivity of the sensor. Right? So these are the factors we can play with. Now coming to the molecular imprinting, why we have chosen molecular imprinting to fabricate cost-effective biosensor? So I'll take a simple example over here. If you this is the object in my hand. If you keep this object and if you mold this object, if you pour a mixture of polymer over this, it will make or it will entrap the whole object, right? And when you will remove this object, it will form a cavity, and this cavity will be complementary to its size, shape, and partial arrangement of the chemical groups, right? So the same chemistry is applied over here. We used Staphylococcus aureus as a template like this. Then we poured the mixture of the polymer, and then we removed the bacteria, right? And then it formed the cavity. Now this cavity can be the complementary to its chemical structure, to its size, shape, right? So this is how molecular imprinting works. We used here polyacrylamide gel because it's readily available, it's cost effective, right? And in any life science lab you can find the polyacrylamide gel, right? So what is the basic chemistry over here, and what we thought that it would work, and why it would work? Because we thought that depending on the size, shape, and surface chemistry is like that. Here you can see polyacrylamide bonds, CONH2 bonds, right? So it will have the specificity based on van der Waals forces and hydrophobic interaction. There is no covalent interaction over there. So it will be easier to remove the template, and it will want to be harsh on the imprinted template. So there is a chance that you may not need any particular chemical to rebind the particular inhalant. Right. So research done so far in this particular area. So here you can see that imprinted technique in which bacterial surface was complexed with exogenous target protein and imprinted silica films with exogenous protein. But here silica gel was used, not the polyacrylamide gel was used. And second thing, it didn't show any reabsorption or say any specificity over the Staphylococcus aureus. Right. So this experiment was published, but it was totally a fail to detect S. aureus. Coming to another objective, they used protein A of S. aureus to imprint a cavity. But again, the method could not be used as a technique for recognition because it couldn't show any specificity. Right. So again, whichever the method we are developing, the ultimate use is it should be commercialized, it should be specific, it should be highly cost effective. Right. So these two methods, these two techniques they developed, but they couldn't reach to the commercialization. Now, reason behind this success of reason behind the less success of imprinting techniques so far. Even the foreign scientists are trying to develop a thinnest polymer which can imprint the whole cells. But using the whole cells, there are some limitations because large molecular size is there, right? So complexity and fragility of the molecule can be one of the limiting factor. Again, imprinted gel bases are difficult to handle while testing the samples due to their shape and size, right? The beads sometimes they are using acrylamide gel beads. the bits applied more for the separation or the removal of the bacteria from the given samples rather than the diagnostic purposes. Right. Instead, gel slab of particular sizes in combination with spectrophotometry make it convenient to handle and test against the target bacteria when it comes to its practical application. So far, using imprinted materials for diagnostic purposes is still a dream come true. So what we proposed here, so here we can say that this is the miniaturized spectrophotometry that we purchased from the market, right? And here this is the gel, polyacrylamide gel, in which we entrapped the S. aureus bacteria and we generated the cavity, which are complementary to the S. aureus, right? And so this is the basic model that we are supposed. And here you can see you can incubate the gel slab against a particular sample, and then you can remove the gel. So before and after, what is the change in the OD? We can measure over here with the help of portable spectrophotometry, right? So which are the applications over here? First is the diagnostic purpose. Our main focus is for the diagnosis. Again, if it is not working over there, then the second option may be food and fermentation industry to remove the contamination. So this is the procedure for the sensor fabrication. So in general, we incubated the bacteria overnight, right? And they incubated at 4 hours to get OD at 1, and we measured it at 600 nanometer. Then centrifugation, everything, right? So after that, I would like to say that we made 4, 6 and 8% of the acrylamide gel. We have chosen such concentration because only in this concentration we could find the entrapment of the bacteria, neither more, neither less. Then we did gram staining to check if the bacteria are really entrapped or not. And if it is there, then we lysed the gel to remove the bacteria with the help of the lysis solution. Then again we did the gram staining to check the bacteria is removed or not. But when we did the gram staining, we could find that the gel was very fragile. So it itself proved that bacteria are removed, and it is very fragile due to the cavities formed inside them, because when we are supposed to restain with the ethanol, right, with the alcohol, it will get shrinked. Right. So incubation of the bacterial cells with imprinted polymer and observation of the change in OD through spectrophotometry at 600 nanometer. Yeah, so this is the composition chemistry that we use over there for 2, 4, 6, 8 and 10%. So as I said, only 4, 6 and 8% showed the entrapment, right? So here, same thing. So here we can observe the gram staining. Right, this is the blank, this is the blank without the bacteria. This is the PAM gel without the imprinting. This is the PAM for 4%. This is the PAM for 6%. This is the PAM for 8%. Here you can see that in 4% the bacteria are clearly visible. So we can say this is the optimum thickness we can find, because if we move towards the if we move towards the 6% of the gel, it is showing more planar orientations. So it is showing hazy bacterial observation, you can see. So we can say that it is thicker, the gel is more thicker, and there is a possibility that it may or may not give the specific results due to the thickness of the gel. Again, 8%, the same results, and even more compared to the 6%. Okay. And when we use the washing solutions, there were two compositions we used according to the literature. 10% acetic acid and SDS, that is 1:1 ratio, and then 0.2 normal NaOH and 1% SDS we used, right? So with the help of NaOH and SDS, we were successful to remove the S. aureus from the gel, right? And then we tried reabsorption with the sample. Here we can see that this is the control, where cells in PBS are showing a particular OD, fixed OD, because it was the cells were in PBS only. There was no media, right? Then 4% of non-imprinted gel here we can see. So there is, after a particular time period of the incubation inside the in suspension, there is a drastic change in adsorption, right? Compared to the non-imprinted. Here when we come to the non-imprinted, right? So up to 45 minutes, up to 75 minutes, sorry, you can see the shift in the reabsorption. So difference of reabsorption of S. aureus on 4% polyacrylamide gel. So here you can see R square is 0.95 for exponential graph up to 75 minutes. Right. So reabsorption in 4% was observed. Then coming to the 6%, the 6% gel was again used in the same manner that we used, right? So here we can see again up to 75 minutes it is showing, but again here we can see the interference. Non-imprinted gel is showing more reabsorption than the imprinted one. So here we can say that 6% is not specific at all for the recovery result. Was again followed, right? Then difference of reabsorption on 8%. Here we can see again at 150 minutes we can see the shift in the non-imprinted gel is more in the change in the OD, right? So it didn't work again. Now we can conclude that 4% is ideal according to our experiment to go ahead with for the detection or set removal, right? So we then try to characterize the imprinted gel, non-imprinted gel, and the imprinted with the adsorbed bacteria with the DSC curve. So what is DSC curve? DSC curve, you are supposed to denature the whole imprinted material ranging from 40 to 140 degrees centigrade. So here you can see you can get the thermograph, like how at each and every degree the imprinted material or say the polymer is behaving. So this is the conclusion over there. This is the main major focus. Non-imprinted 4% PAM gel showed smaller thermogram value compared to 4% imprinted PAM gel before and after adsorption. Due to hydrogen bonding between the reabsorbed Staphylococcus aureus and polyacrylamide gel network, right, explains that Staphylococcus aureus cells were effectively adsorbed onto the imprinted gel surface, leading to higher thermal stability. Imprinted gel showed higher thermo values compared to non-imprinted, as not all of the targets get completely removed from the gel, and some of the bacteria still may be there inside the cavities, right? So during the adsorption process, imprinted sites are again selectively occupied by the bacterial entities. As well as bacterial surface offers many hydrogen binding sites through its various surface proteins and lipids. Thus, increased binding of bacteria to the gel surface suggests imprinted gel showing high adsorption. These are again the studies which are supporting our results. In contrast, non-imprinted polyacrylamide gel showed non-specific adsorption due to lack of imprinted cavities. Right, so here we can interpret that 4% is showing adsorption, right? Now the dynamic adsorption of 4% imprinted polyacrylamide gel by different bacteria over a period of 75 minutes. We want to check the specificity if SOS is only getting adsorbed or another bacteria is also getting adsorbed at the same specificity, right? So we tried Salmonella typhi and E. coli against S. aureus, and here we can see that S. aureus is showing specificity with the highest change in the OD, right? So it is somewhat specific, somewhat not specific, we can say, because it is all again showing Salmonella typhi and E. coli at some extent they are interfering in the results. Right. So here we can see that OD change was higher in S. aureus compared to S. typhi and E. coli. Right. So then we wanted to check how much bacteria are actually removed or say adsorbed, right? So for all the S. aureus, S. typhi and E. coli, we plotted a graph using colony count method, right, to check how much bacteria is there against the OD. Right. So this is S. typhi colony count method, E. coli colony count method. So change in the optical density of the bacterial suspension before and after adsorption on 4% PAM. Here we can say that OD changes for S. typhi, S. aureus, E. coli are for S. aureus it was 0.1094, S. typhi it was 1094, E. coli it was 1094. This is the initial OD. Sorry for that. This is initial OD, and then the relative CFU, relative change in the OD. You can see here, right? So absolute relative CFU we could found at the last was 5.7633 for S. aureus. For S. typhi it was 5.134, and for E. coli it was 11.42. Right. So here we can say SOS is not showing that much reabsorption, but yeah, when it comes to change in the OD, it shows the highest change in the OD. Right? So somewhat it is specific, but when it comes to the counting the bacterial cells, it is not showing that much reabsorption. So so coming to the conclusion of this graph, over a period of 75 minutes of incubation, the adsorb S. aureus, S. typhi and E. coli bacteria were calculated as 5.763 10⁶ CFU per ml, 5.13 into 10⁶ CFU per ml and 11.42 into 10⁶ CFU per ml respectively. The results demonstrated that surface molecular imprinting is effective for detecting the presence of specific bacteria in the sample, but how much specific, that we still need to identify, or we still need to figure out. Right? As a concluding remark, it can be said that the idea of whole molecular cell imprinting to detect the target bacterial cells seems to be far more from the commercialization. Now coming to the summary. Fabrication chemistry, as we know, it was PAM, polyacrylamide gel only, right? Analyte was S. aureus. Response time was 75 minutes. Specificity somewhat specific, somewhat not. We are still supposed to figure out. Focus of the work was proof of concept only. We are not at the stage of R&D or miniaturization. Right? A type of detector, it was spectrophotometer, so obviously it is optical. Right? Detection limit we could found was 5.763, 5.30 and 11.42 into 10⁶ CFU per ml respectively for S. aureus, S. typhi and E. coli. So intended applications may be diagnostics and removal of the contaminant in the food industry. Right. So coming to the final concluding remark, design of the novel biosensor, it is novel biosensor because no one has reported such work so far. Troubleshooting of the issues faced by the already published work or the conventional method, as I say, ELISA, whether it is ELISA, RT-PCR or colony, this method has some advantages over it, but it is not yet fully approved or fully proved, right? So whole cell molecular imprinting for Staphylococcus aureus proved specificity based on the surface chemistry and shape of the organism. What can be the future prospects, or what can be the steps further need to be taken? Further R&D is needed to check cross reactivity with other organisms with same size, shape and detection limits. One thing, and R&D for miniaturization is recommended. These are the references. Coming to the acknowledgement, since this is my PhD work, I would like to thank my mentor, Professor Nathu Ram Puri, head of the department, biochemistry. Then PhD research fellowship to the MS Institute of Baroda, and Meds of Southern Biomedicals to give me a room to present over here. Right. So, thank you.
Thank you, ma'am. You were the youngest speaker in our Artemis 2024.
Thank you.
Thank you, ma'am, for engaging our session with an interesting piece of work. I now request Dr. Satyendra Mishra to please present a memento to our speaker, Dr. Jinal, as a token of love and appreciation. Thank you so much. A special mention to all our speakers for completing the sessions very timely. Thank you so much. So, we hereby end up with all our technical session, rather technical expert sessions, which was scheduled for this meet. And as arranged, we will now disperse for lunch, which is arranged at Central Lawn, where we had our breakfast. Before that, a small announcement to all the poster presenters. Please, before you move towards your lunch, please put up your posters. We have our volunteers there. And at sharp 4, we will meet again at the Central Lawn for our valedictory, where we will be having our prize distribution ceremony as well. Yes. And we also have a QR code which is given in your iCard. Please scan that and put your feedback. This really means to us. So we all will see you at 4 for valedictory session, and at sharp 2 we will begin our poster presentation evaluations.